Hello friends, welcome to Neat with Bioscience. Today we will continue our discussion on Krebs cycle. Yeah, up till now we had seen that Krebs cycle is the central metabolic pathway. How? Because the fat, carbohydrate or protein or amino acid, they all will undergo the individual metabolic process or their individual catabolic pathway, and ultimately they will convert to acetyl CoA, and that acetyl CoA, this acetyl CoA will then enter into Krebs cycle. Yeah, so fat will also undergo its individual metabolic pathway, carbohydrate, amino acid, and then they all will come to one common point, that is acetyl CoA, and this acetyl CoA will then enter into Krebs cycle. Yeah, in my last class we had seen all the reaction of Krebs cycle, which is a 8 enzymatic steps. Yeah, today we will continue our discussion on Krebs cycle, about the regulation of Krebs cycle, inhibitors of Krebs cycles, and amphibolic role of Krebs cycle. Amphibolic means Krebs cycle is both catabolic as well as anabolic, right? So that is known as an amphibolic role. So today we will discuss all these points. So let's start our discussion. So first, the quick review of Krebs cycle. As we had already mentioned, Krebs cycle, acetyl CoA will enter into the Krebs cycle. So first, acetyl CoA, which is a 2-carbon compound, first it condenses with oxaloacetate, which is a 4-carbon compound. So 4-carbon compound, 2-carbon compound in the presence of enzyme citrate synthase. Okay, citrate synthase in the presence of enzyme, and one water molecule will add here. So this coenzyme will remove. So acetyl moiety will condenses with oxaloacetate and we will get the citrate, right? Citrate, which is the first product of Krebs cycle, 6-carbon compound. Therefore Krebs cycle is also known as a citric acid cycle, and as this citrate has a 3-carboxylic group, therefore it is also known as a tricarboxylic acid cycle. Yeah, then citrate will isomerize into isocitrate. First there is a removal of water molecule and then addition, means dehydration and rehydration in the presence of aconitase enzyme. So there is a just shifting of the OH group, nothing else. Both are 6-carbon compound, but the position of OH group will shift from one carbon to another. So citrate will convert to isocitrate. Then this is important step. Isocitrate will convert to alpha ketoglutarate. Here the 1 carbon dioxide will blown out here. Yeah, acetyl CoA is a 2-carbon compound. No? So out of that 2 carbon, first carbon will blown out here, and as there is a carbon-carbon bond breakdown. Yeah, so energy will be captured by NAD. So there is a production of one NADH plus H positive molecule. Isocitrate dehydrogenase is the name of enzyme. Okay. Second carbon dioxide will be removed during the conversion of alpha keto. Yeah, this isocitrate will first convert to alpha ketoglutarate, 5-carbon compound, and then alpha ketoglutarate will convert to succinyl coenzyme A. Yeah, succinyl coenzyme A. So there is a decarboxylation, second carbon will blown out, dehydrogenation. So there is a formation of NADH plus H positive, and there is an addition of coenzyme A. So we will get the product succinyl coenzyme A. Then succinyl coenzyme A will convert to succinate. Enzyme is succinyl coenzyme A synthetase. Succinyl CoA synthetase. Okay. Yeah, and succinyl coenzyme A will convert to succinate, and this there is a removal of this coenzyme A. During this step, GTP will convert to GDP and we will get direct ATP, means ADP will convert to ADP, direct substrate level phosphorylation. So we will get ATP here. Yeah, after that, once we will get this 4-carbon compound, then all the product till oxaloacetate will be 4-carbon compound. Okay, then succinate will convert to fumarate. Enzyme is succinate dehydrogenase, and there is a production of FADH2 instead of NADH. All dehydrogenase in the Krebs cycle will produce NADH. All dehydrogenase will produce NADH except succinate dehydrogenase, which will produce FADH2. Yeah. So succinate will convert to fumarate, then fumarate will convert to malate, enzyme is fumarase, and finally malate dehydrogenase enzyme is there. So malate will again come back to again we'll get our original product of Krebs cycle, that is a oxaloacetate, and one more NADH will produce here. So total 3 NADH, 1, 2 and 3 NADH and 1 FADH2. 3 NADH, 1 FADH2 and 1 ATP, that is direct ATP. We can use it, we can call it as a cache. Okay. Now we start our topic, that is the regulation. Okay. Regulation of Krebs cycle, it depends upon the energy status of the cell. Energy status of the cell. Yeah. So what will determine the energy status? ATP, NADH. How NADH? Because NADH will then enter into the electron transport chain, and from 1 NADH we will get 3 ATP, right? So ATP, NADH, these are the compound which will determine the energy status of the cell. And if there is a large amount of ATP is there, NADH is there, that means energy status of the cell is high and the cell is in a rich energy state. So we need more of Krebs cycle or less of Krebs cycle? Yeah, we need less of Krebs cycle because energy status of the cell is sufficient. In that case, Krebs cycle will be slowed down. Okay, and if ADP is there, ADP, because we know ATP will hydrolyze into ADP and phosphate group. So if ADP is there, NAD is there, yeah, there is a large concentration of ATP, NAD, yeah, these are the indication that the cell is in a poor energy state. So in that case, we need more of a Krebs cycle. 3 enzymes which are mainly involved in the regulation of Krebs cycle. The first is this citrate synthase. Okay. Second is this isocitrate dehydrogenase, and the third one is this. These 3 enzymes are involved in the regulation of Krebs cycle. Okay. Now let's see how. Citrate synthase, isocitrate dehydrogenase and alpha ketoglutarate dehydrogenase. Okay. This will determine the rate of Krebs cycle. Okay. So 3 enzymes are citrate synthase. Okay. Second is a isocitrate dehydrogenase, and the third one is a alpha ketoglutarate dehydrogenase. These 3 enzymes, these are important for the regulation of Krebs cycle. Okay. As we had already mentioned, if ATP is there, so ATP, NADH, it will regulate the enzymes via allosteric regulation. Okay, allosteric regulation means apart from active site, one more site is there where the product will bind, which will either increase or decrease the rate of enzyme activity. Yeah. So ATP, NADH, these are the, if this substance are present in a large amount, that means energy status of the cell is high. Yeah. So we need less of Krebs cycle, that means these are negative regulator. Negative regulator. Okay. ATP, NADH. So all the 3 enzymes will be negatively regulated by ATP, NADH if this substance are present or this compound are present in a large concentration. ATP, NADH. Okay, these are negative regulator. Citrate synthase also negatively regulated by if high concentration of citrate is there. See, oxaloacetate will convert to citrate. So this is the product of this reaction. If this product is present in a large concentration, so it will regulate the enzyme activity and this conversion will not take place. So ultimately Krebs cycle will not run further. Okay, so citrate will also regulate this enzyme citrate synthase. So if the citrate is present in a large concentration. Okay, and one more product of the Krebs cycle was succinyl coenzyme A. So this succinyl coenzyme A will also, because see, yeah, and here we will get a succinyl coenzyme A. If we enough of succinyl coenzyme will accumulate, so this will also give the signal to citrate synthase. So much of succinyl coenzyme is there, so that will also negatively regulate the enzyme activity. Okay, that is known as a feedback inhibition. So via feedback inhibition, succinyl coenzyme A will also inhibit. So these four are the negative regulator of citrate synthase. Okay, isocitrate dehydrogenase has ATP and NADH. Alpha ketoglutarate, yeah, this alpha ketoglutarate. When alpha ketoglutarate will convert to succinyl coenzyme A, yeah, that was the step in the presence of enzyme alpha ketoglutarate dehydrogenase. So succinyl coenzyme A, like citrate will inhibit the activity of, if the more citrate is there, so that will inhibit the activity of citrate synthase. The same way, if the more succinyl coenzyme A, so it will also negatively regulate alpha ketoglutarate dehydrogenase enzyme. So succinyl coenzyme A will also act as a negative regulator of this enzyme. Succinyl coenzyme A. Okay, so these are the negative regulator. 4 for citrate synthase, ATP and NADH for isocitrate dehydrogenase, ATP, NADH and succinyl coenzyme A, yeah, succinyl coenzyme A via feedback inhibition. So these three are the negative regulator of alpha ketoglutarate dehydrogenase. Now we will see the positive regulator. Yeah, so reverse will be the case. If ATP is there, so that will act as a negative regulator. And if ADP, adenosine diphosphate, if ADP is there, means we need a more of Krebs cycle. So that is act as a positive regulator. Okay, so for citrate synthase, ADP, okay, for isocitrate dehydrogenase, ADP and calcium ion. Okay, how calcium ion? If we take the example of muscle cells, so during the contraction, yeah, if the muscles are working, so during the contraction of muscles, there is a large influx of calcium ion into the cell. So when there is a large influx of calcium ion, at that time ATP hydrolysis will take place because work is being done over there. Okay, so ATP hydrolysis, so ATP will convert to ADP and PI. That means the more of ADP will be there. That means we need more of Krebs cycle, yeah, or more of enzyme activity. So presence of calcium ion will also act as a positive regulator. Means the muscle cells are working high. Therefore, calcium ion will act as a positive regulator, and specifically for muscle cells. Okay, and this calcium ion will also positively regulate alpha ketoglutarate dehydrogenase. Okay, so these are positive regulators and these are the negative regulators. Okay, now next we will see the inhibitors. Inhibitors of Krebs cycles. You know, those who are into the research field or yeah, means they are doing some research or they want to control the enzyme activity or the control the cellular process. So these are the point where the regulation, inhibition takes place. For those students, these points are very important because these steps are under our control. We can manipulate these steps. So regulation or inhibitions, though these points are important for research purpose. Okay. Now inhibitors of Krebs cycles. There is Krebs cycle. Okay. You can remember like F A M A K S. First we will see the enzymes. 3 enzymes are there. If we add some substance on it, or if we put some reactant over it, so it will bind with the enzyme activity and that will completely inhibit or stop the Krebs cycle. Yeah. So that way, see, like we will see here, aconitase enzymes. A is for aconitase. Yeah. And inhibitor is fluoroacetate. So this acts as a rat killer. Yeah. So if fluoroacetate is there, it will completely inhibit the activity of these aconitase enzymes. Yeah. So that means the Krebs cycle will be stops, and ultimately we'll know the result. There is act as a rat killer. So what will happen? Okay. So this way we will inhibit the Krebs cycle. So aconitase, this for alpha ketoglutarate, dehydrogenase and succinate dehydrogenase. These 3 enzymes are theirs, and inhibitor for these 3 enzymes are for aconitase, fluoroacetate, alpha ketoglutarate, arsenite, and for succinate dehydrogenase, malonate act as a inhibitor. Okay. These are Krebs cycle. And now the third point we will see is amphibolic role of Krebs cycle. Amphibolic role. Amphibolic role, that means both catabolic as well as anabolic. So Krebs cycle is both catabolic as well as anabolic, because see, this acetyl-CoA will enter into Krebs cycle and will get carbon dioxide, yeah, and NADH. So that will use as an energy generator. So this is known as a catabolic step. Okay. And some of the intermediate will involve in the synthesis of some products. So that is anabolism. So Krebs cycle is both catabolic as well as anabolic. Therefore, it is known as a amphibolic role. So we'll see how. First is oxaloacetate. So oxaloacetate will involve in the synthesis of aspartate. Yeah, and this will ultimately involve in the synthesis of purines and pyrimidines. Purines and pyrimidines. Second is a citrate, and this is very important, this citrate, because most of the MCQ there are many questions, multiple choice questions about this citrate too. That citrate will convert to acetyl-CoA in cytosol. So first citrate will come out, because see, we know that this Krebs cycle will take place in the matrix of mitochondria. So citrate first will come out of mitochondria, and in the mitochondria citrate will convert to acetyl-CoA, because acetyl-CoA cannot cross the mitochondrial membrane. So citrate will come out, citrate will convert to acetyl-CoA, and this acetyl-CoA will then involve in the synthesis of fatty acid. And cholesterol. fatty acid and cholesterol. Then oxaloacetate, citrate, then alpha ketoglutarate. This will involve in the synthesis of glutamate and ultimately into GABA, that is gamma amino butyric acid. Okay, and the last is succinine coenzyme A, which will involve in the synthesis of heme and ketolysis. Okay.